Laboratory tests take place at all levels throughout our biology. There are all kinds of body components that get poked and prodded and tested. This video attempts to explain where applied diagnostics testing fits into that healthcare continuum. What you have in front of you is a cell. Cells have these antigens, or character-defining proteins, on the surface of the cell, expressing the nature of that cell. Some cells have certain kinds of antigens, and other cells have different antigens. In the case of bad cells, the antigens would indicate that they are bad cells. The body produces antibodies that are floating around seeking an opportunity to find bad cells and to bind to them. The human body produces tens of thousands of antibodies. Antibodies are constantly on the prowl for cells with antigens that fit their specific antibody. In the case of our bad cells, when they find a match and bind, much like a magnetic attraction, they get carried off together by other cells that specifically search for a bigger cell and pull them out. With all our diagnostic platforms, we use antibodies and markers to decorate the outside of the cell, or many cells, the cell interior, inside the nuclei, and inside chromosomes to personalize diagnostics down to the DNA level. When you use one or a few antibodies at the same time on a few cells, the process is called immunohistochemistry. And that is where our tour of applied diagnostics technologies can begin. But for us to analyze at the cellular and subcellular level, we must start with a specimen. The specimen consists of a clot, core, bone marrow, including spicules, and peripheral blood. Here you can see how specimen pieces correspond with testing platforms used to process it. At the beginning, all of these platforms are accessioned and triaged, meaning as these cases come in, experienced laboratory personnel identify which cases are critical, which ones move to the front of the line, which ones might be medically stat and processed accordingly. Immunohistochemistry, or IHC, is primarily done on solid tissue, but there is a smear preparation, represented in this slide, that is completed where blood is spread out over a slide. Then it is examined to see under a microscope what might be going on morphologically. The steps for morphology are as follows. Step 1, there is grossing, which is the description of what the specimen looks like at the macro level. A macro description might include definitions of the size in centimeters, weight, or color, perhaps dark brown. Again, gross descriptions. Next is histology. 3 steps in histology are: the specimen is embedded into a paraffin block. Then slices are shaved off of that paraffin block, and the slices are applied to a slide and are stained with H&E. These are the principal stains in histology. Then the next step is immunohistochemistry. That's the step in which there are markers applied to the slide and then looked at under the microscope. There you have the end result, and a morphology that is done by day 1.5. Similar to immunohistochemistry, flow cytometry uses markers to characterize cells, but does so with more markers and more cells. In simplest terms, flow can take a mishmash of cells and define them. Flow cytometry looks at multiple markers on tens of thousands of live cells floating in a fluid-based medium. The process is automated and offers results within hours. This is called flow cytometry. There are 3 quick steps here. Step 1 involves preparation, where tubes are set up in such a way to include specimens. They're incubated, red blood cells are removed, and antibodies are added. In step 2, those tubes are run on our flow cytometer to detect decorated cells that are now illuminated by antibodies. In step 3, from the machine we get data that presents characterizations of cell populations within a larger volume of tested cells. Now, if we move further into the cell towards the nucleus and study chromosomes, you're in the area of cytogenetics. And by cytogenetics, we mean that we look at the chromosomes, take a picture of those chromosomes, and then separate them. We arrange them into pairs in descending order and find abnormalities if they exist. There can be abnormalities in terms of the number of chromosomes. There are normally 23 pairs of chromosomes. Sometimes you have an extra one. There could be 3 chromosomes instead of just 1 pair. So you have an extra chromosome. That's abnormal. Sometimes a piece of the chromosome can be missing and attach itself elsewhere. That's an example of an abnormal structural issue. Cytogenetics is the longest and most involved process that we do at the laboratory. It begins with wet lab processing, where cells are incubated, grown, and harvested. They may be grown for a period of 24, 48, or 72 hours, depending upon the kind of case. Then those cells are harvested. Post-harvesting, we drop cells onto a slide. This term comes from an old method of dropping cells onto a slide from a high elevation to create a spread, much like dropping a water balloon. Now, with better technology, we fix slides that disperse the cells with heat and humidity to control the cell spreading process, offering a good spread of the chromosomes. Then, they're scanned overnight and are finally analyzed. That means all the chromosomes, the pairs of chromosomes, are identified, they're isolated, separated, and arranged in descending order from largest to smallest, a process we call karyotyping. Finally, abnormalities are identified and ultimately reviewed by the cytogenetics director and signed out. That concludes cytogenetics. If we continue to go deeper into the chromosome, we can then study abnormalities in the sequence of genes. Genes code proteins that create our individuality. With genes, there are certain rules. T only binds with A. C only binds with G. We also know that certain sequences of these genes mean certain things. One sequence means blue eyes, while another sequence means blonde hair. Another sequence will mean resistance to a particular type of chemotherapy, or it could mean a certain type of disease. When we know what that sequence is, and we want to identify it, then we can make the complement to that gene sequence, paint it with a fluorescent probe, and then send those probes looking for those sequences. We see the sequences and count the probe lights. This is essentially the way molecular and FISH work. FISH actually begins with the wet lab processing of cytogenetics, as we borrow cells straight from that process. From there, FISH prepares slides, separates DNA bonds, then sends probes on a quest for their targeted sections of DNA called base pairs. Probes attach themselves, and you have DNA that is tagged by a probe. They are then analyzed by essentially counting the probes using a fluorescent microscope, and that is FISH in a nutshell. With the molecular approach, we're analyzing DNA from the nucleus of the cell. DNA sequencing technology allows us to identify the order of targeted bases, for example, A, T, C, or G, to determine any presence of mutations in the gene sequence. The steps in this process are as follows. Step 1, we extract the DNA. Step 2, then amplify the DNA by first breaking DNA bonds, similar to the science in FISH. We then bind targeted primers with organic elements that naturally extend or copy those sequences and repeat, repeat, repeat. This process of copying sequences gives us greater visibility into a patient's DNA, and that's why it's called amplification. Step 3, the DNA is sequenced and analyzed and then reviewed by a bioinformaticist so that the results can be clearly relayed to the physician. Collectively, physicians use that data to apply the most personalized approach to patient therapy. Sendouts are shipped, tracked, and reported as needed. From thousands of cells to specific DNA sequences, Applied Diagnostics is operationally structured around providing the deepest possible insight for partner physicians and their patients.